So thanks, Michelle. This is Stuart, and I also have in the room with me Jean-Noel Biaud, who the two of us worked on this case study together. And also on the phone, we have Sean Prince, who is a field application scientist for CLC Bio. So as we talk about that, if you have questions that are in depth about the CLC workbench, let us know, and he can answer those. So just to give you a brief agenda, we're going to talk about IPA as a case study of host response to infectious disease. I'll go through a brief background on IPA, and then background on the West Nile virus, and then this case study of the macrophages from humans infected with West Nile virus and measured by RNA-seq expression. And then we'll also compare it to a similar infection but in a different cell type and using microarrays as opposed to RNA-seq. And also, as we go along, I'll hope to highlight some of the newer features in IPA that have come out recently. So a little background on Ingenuity or IPA. So it's been around about 11 years, IPA has, and 2013 was our 10-year anniversary. There are something we're proud of as a company is just how many people have published using IPA. So in the methods or in the paper, they'll mention that IPA was used for the analysis of data, and that's over 12,000 publications now. And in 2013, we joined QIAGEN, as did CLC Bio, which we'll talk about in a minute. So let me give you a little background on what we do, what Ingenuity has been doing for the last more than actually 13 years, is acquiring findings or facts from the literature and other sources. And we have PhD scientists who read journal articles and curate findings from the articles and structure them in a way that is useful to do analysis with those findings later computationally, because we understand how biological concepts are connected to one another in an ontology. And so we have over 5 million findings now in our knowledge base, and those are used in IPA to analyze data, and I'll show you in great detail how that works in the next few minutes. So just to help explain ontology, so it's just this is an example of the ontology for the replication of flaviviridae, and you can see how it's structured into this hierarchical kind of thing where it's an RNA virus, it's a viral infection, and things like that. It's part of the disease. Things can have multiple inheritance, so there could be a replication of flaviviridae could be classified under some other parent thing as well. But in this case, you can see where it sits, and then underneath that is the replication of West Nile virus. So when the knowledge curators go to the literature and read something about a West Nile virus paper, they understand how to connect it into the ontology. So what is a finding? So this is an actual finding in our knowledge base that's relevant to this talk. So it's about fibroblasts in a certain type of stage of mouse, embryonic. It's a homozygous mutant RIG1 gene. If you knock it out, it increases the rate of infection of fibroblasts in cell culture by the virus, West Nile. And so there's a lot of really good information in here, such as the cell type, the species, the specific gene, the type of mutation, the development stage, the zygosity, what agent was there, the biological function it affects, and most importantly to us is the direction of the effect, in a sense. That's the thing that says what happens in a directional way, which is more valuable than just it's associated with this, knocking out a gene is associated with West Nile virus. It increases the infection. And so these are used computationally when we analyze data, or when IPA analyzes data. So, and one of the other things we can do is infer things like the activity of the molecules decreased. So there's another type of direction in a sense. So loss of the activity increases the infection. Okay. So let's go into a little bit about West Nile virus. So it's a member of the flaviviridae. It's a neurotropic virus. It's in the same family as yellow fever and dengue. It's a insect-borne virus. It causes encephalitis, viral encephalitis, around the world. It's not completely understood how that happens. There's some ideas, but it's not completely understood. Here's some details about the structure of the virus, and then here's the sort of just a diagram of how it's really it's a bird virus, but it tends to accidentally affect humans and horses, and probably other animals. Okay, so there's no approved therapy for it, and it's a real problem. You know, there's several thousand cases a year. Normally it doesn't really have a bad effect on people, but it can be severe in elderly and immunocompromised, with neurological effects and effects on the eye. It crosses the blood-brain barrier and can cause damage to the eye and to the brain. It's characterized by neuronal death and the immune cells of the host potentially causing the damage that's in the brain by trying to clear the virus. The immune response is interferons of type 1, then the humoral immunity, as well as cellular immunity, which helps recover, to clear the infection and recover. It can be shown in mice. It induces a systemic infection and does actually cause the same kind of phenotypes as in human. Depleting the mice of certain immune type cells actually causes a longer, extent of the viremia and increased death by the virus. So the immune system is blessing and a curse, trying to resolve the infection, but can also cause damage. So we wanted to analyze this data in IPA. What can we learn from expression data of the host? So we went to the literature and found this paper and this study, which looks at, again, primary macrophages from healthy human donors. There's 10 people, and the multiplicity of infection was 1 for the virus, so not a heavy dose of virus, for 24 hours. We took this FAST-Q data and put it through the CLC Cancer Research Workbench to process the RNA-seq data, and I'll describe that in a second here. So just a little bit about the CLC Cancer Research Workbench. It's a fast and customizable tool. I think that Jean-Noel and I can both attest to it being pretty easy to use. This is our first experience with a tool to process this kind of data. It was pretty quick to understand how to use it. It's end-to-end analysis workflows are available. You can have single samples or cohorts. It works for more than RNA-seq. I want to make that clear. We used it to generate RNA-seq data, but it works for all kinds of variant calling and panels like from QIAGEN, GeneRead. And the cool thing for us especially is it has a streamlined connection to IPA. So you can process the data in the workbench and then just click and send it to IPA. So it's really easy. Again, you can do a whole different variety of kinds of omics analysis, and you can make workflows that can be repeatable, and you can have a sort of a pipeline. And you can look at even 3D protein structure and see how variants fall on the three-dimensional structure of a protein, which is really cool. It's got a genome browser, and it does a lot of things. It's very nice. And so in simplified steps that we did was, again, take that data set, download the FAST-Q files from either the European Nucleotide Archive or from SRA. Just as a point, European Nucleotide Archive is a little easier because the files are already in FAST-Q. With SRA, you have to process them into FAST-Q files. So that saves you a step there with European Nucleotide Archive. So you import the FAST-Q files into the workbench, which I'm going to call CRWB here, and you set up this analysis, which means mapping the reads to the reference genome, which is human, and you're quantifying transcripts, essentially. And then you create an experiment where you actually ratio the, mock versus, in this case, mock versus the West Nile virus infected, and you compute stats for the p-values and false discovery rates and things like that. And then, like I said, you just quickly send it to IPA using a plugin, which you can download from CLC. And we're sending this fold change, the p-value, and the false discovery rate, the q-value. So long story short, for our case study, we used ensemble-based mapping at the isoform level. So these are individual transcripts per gene. And the data set ended up, we got 3,285 isoforms when we said we must have a greater than 10 RPKM in either one of either the mock group or the infected group, and a fold change at least greater than, absolute fold change greater than 1 and a P less than 0.05. So that's the data set. And then in the IPA analysis level, you fold it to the gene level, and we filtered more strongly. So 893 genes passed an absolute fold change cutoff of greater than 2 and a false discovery rate of less than 0.05. So that's the 893 genes that went into the analysis. But we have the isoform level information in there as well, and I'll show you how you can use that in IPA. And that's sort of, some of that's newer, the newer features. So you run a core analysis, as we call it in IPA, and you get a set of results, and they're in tabs, and they are, the canonical pathway predictions. Are they increased or decreased predictions for activation of the pathway or inhibition? What upstream regulators might be activated or inhibited to affect the expression changes that you see in the data set? The diseases or the functions, we call them biological functions or biological processes that are predicted to be increased or decreased in the set based on the fold changes or based on the gene expression changes in the data set, and then how upstream regulators may drive those diseases and functions and phenotypes. This is an integrated analysis of the upstream and the downstream together, which we call regulator effects, and I'll show that. And then there are things like how just interaction networks, how the molecules may interact with each other. Actually, I'm not going to show that. And then you can even have your own pathways, your own gene lists, and you can determine the overlap to your data set statistically. And I'm not going to show that either. Okay, so which canonical pathways are activated or inhibited? So this is a screenshot of the results in IPA, and so you see how EIF2 signaling is predicted to be decreased. This is about protein translation, which is known as a thing that can be decreased during a viral infection, which just makes sense. So blue, just to say blue, this is going to be a theme throughout IPA. Is blue means decreased prediction, orange means increased prediction of increase. Okay, so interferon signaling is predicted to be increased in activity. So this pathway is predicted to be activated. And you can see a whole number of other pathways around dendritic cell maturation and other pathways that are affected or increased by prediction by this data set. So let's delve into the interferon signaling though a little more deeply. So this is the pathway, and what you see are the pink ones are up, or pink or red are upregulated in the data set. The yellow highlight, I won't go into too much right now, I'm going to go into it a little more later, but it indicates that there's more than one isoform in the data set. So meaning there are multiple isoforms for these yellow circled ones. And we'll go into that in a minute. But so you're seeing the RNA-seq information here, so you can dig into that. And so lots of genes are activated or upregulated in this data set that map to the interferon pathway. One thing you can do is add, well, what's the biological function of those genes? We know it's an interferon, but is there a more specific level of information we can add? And so in IPA, this is sort of a recent feature, you can add, statistically figure out what functions or biological processes are relevant to this set of molecules. And so we did that and found that antiviral response and replication of flavopurity are relevant, and then we added them live to the pathway. And what's cool about that is you can then simulate what would the effect of the changes in the expression of these genes on those functions. So it's called the molecule activity predictor, and it predicts, and again the orange and the blue, it predicts that there's an increased antiviral response and decreased replication. So the cells are fighting the infection using these molecules. They're activating these molecules, which in the interferon pathway that have this antiviral response essentially. So the next topic is downstream effects. I showed you two things. The increased, I just showed you a couple functions you could add in a sort of user-directed way, but you could also just ask about all functions at once in an unbiased way. What's going on here downstream? And what you see is what we call the downstream effects analysis. It's a hierarchical heat map. So each major box is a category of functions, like cellular movement, like immune cell trafficking, infectious disease. And each little square is a specific type of immune cell trafficking or a type of cellular movement. And the color, again, is an indicator of the prediction. So you can see how there's a lot of immune cell trafficking prediction of increased cell movement, cell-to-cell signaling. And then below this is a table which shows you the actual specific results. So you can see how the development of lymphocytes is increased. And it's just an obvious strong immune response that's predicted from this gene expression changes. You can drill down into all of these squares and look at them in more detail, but just in the interest of time, you can also do things like you can click on a couple of these, or however many you want, and show them as a network, because these are essentially a function is just a set of genes connected to their function, what they do. So this is picking 3 of them, and then you might not be able to see them so well. It's antiviral response of cells, the recruitment of lymphocytes, both predicted to be increased. So again, increased antiviral, the cells are recruiting white blood cells if there were any in the culture, or they're macrophages in this case, and then decreased replication. And you can, this kind of view, you can see whether there's common genes between them. Like, so you can see that interferon beta 1 has, is involved in both antiviral response and replication of the virus. Another thing we have that's new, though, is a new layout menu, which allows you to change the layout of these molecules on this, or these nodes on this network, and you have these various choices. What's kind of neat is you can do a different one, like hierarchical, like this one, and I didn't do anything. I just clicked that button, and then it put CD40 at the top. So, and that's because it's connected to all 3. And what that shows you, though, is that CD40 can drive all 3 functions. It might have been not as easy to see that in this view, and so that's kind of a nice tool to learn something from the structure of the network that might not be obvious to your eye in a different layout. Here's an organic layout, which packs the molecules a little differently on the map and makes it a little easier to see sometimes. So it helps in publication and things like that. And also you can discover something you might not have noticed with these layouts. So that's a new feature. Okay, so, and I just want to emphasize that I talked about the knowledge base and how you can, everything's backed by these papers that people have read, scientists have read. You can double click on any line to figure out what is that relationship. And so here's a case where this IFITM3 protein, it connects to replication of the West Nile virus through, here's the paper that backs that finding, and et cetera. So it's always available, the literature behind things. Okay, so let's do a little bit of how things work. So how do you predict upstream regulators in the data set? So the way this works in IPA is that it's kind of, think of it as a pattern match. So we have in the literature, from the literature, we have upstream regulator, which could be any molecule. This could be a microRNA, it could be a transcription factor, it could be a kinase, some molecule that has some effect on the expression of other genes. And this is all from the literature. And so we have an expectation. In this case, this generic upstream regulator is supposed to turn on the expression of these ones, and it's supposed to turn off or inhibit the expression of these 2 molecules. These are meant to represent the molecules in your data set. And so from the literature, we have a certain expectation as to what they should do if this were activated. The algorithm looks at the actual expression in the data set and determines if there was a match, meaning the upstream regulator looks as if it were activated, or an anti-match where everything went backwards, let's say, predicting that upstream regulator is actually inhibited. Okay, so that's how the tool works. It uses a Z, and it comes up with a Z-score, and a Z-score is greater than 2 or less than -2 are considered significant. So that's how that tool works. And what you and you can then filter the results by the type of upstream regulator. So in this case, we filtered the table to only look at cytokines as upstream regulators. So you can see this strong interferon response, for example. Or you can say, I only want to see the transcription factors that are activated. And here are a bunch of interferon-regulated, or regulated transcription factors that, and other immune response type of transcription factors. So this is something that's easy to do, and you can learn just by looking at this set of molecules, you can understand what might be going on biologically. You can also do this integration, like I talked about, which is upstream plus downstream, which we call regulator effects. So the idea is, if you have an upstream regulator, as I just described, and you have a disease or function, as I described earlier, they're both using molecules that might be in common. So an upstream regulator might be predicted from this set of 5 data set molecules, and the same 5 data set molecules might also impact a function. So you can statistically merge those two together and find cases where you think the upstream regulator might be able to explain that, or the disease or function's output might be explained through this upstream regulator working through the data set molecules. So this is the kind of networks that this tool in IPA just automatically creates for you. And sometimes it is a case where we know in the knowledge base, it's known in the literature that an upstream regulator has an effect on the disease or function. If such a connection is known, it'll be shown to you in IPA, but it doesn't require that. So you can find novel upstream regulator disease function relationships, via sort of bootstrapping via the data set targets. And so it also it runs iteratively and connects multiple upstream regulators and multiple disease or functions. So this is a simple one, but it actually puts more regulators and more disease or functions together to have a sort of overall hypothesis. So here's a case where we've just filtered the results for transcription factors. So upstream regulators must be transcription factors in this network, and the functions must be infectious disease functions. And so you get a network like this where there's multiple transcription factors that work together, and you can see there are even connections between them. IRF7 has an effect on IRF1. Working through these data set molecules to inhibit replication of the virus. So this is a hypothesis for how these transcription factors do their job. You can also put other types of networks together. You can change the algorithm and run it again, which is what we did here. So now we're saying only show us microRNAs and their connection to any biological function. And you can see how this is, again, the blue predicting that miR-146 may be inactivated or downregulated in the data set. We didn't measure, we don't have a measurement of miR-146, but if it were regulated, it would be down. And it's affecting potentially these data set molecules. So by virtue of miR-146 being downregulated or inhibited, you get this effect on these molecules in the data set, which then in turn drive potentially increase of synthesis of nitric oxide, cell movement, things like that. So what's kind of interesting though, if you go to the literature, there is a case, you can see a case where miR-146 loss may be associated with autoimmune conditions. And so from the paper it said it implies that loss of that microRNA may lead to an auto-inflammatory condition and cause exaggerated inflammatory response. And so this may be a hypothesis for how the virus can cause an over-exaggerated immune response, which could lead to the encephalitis or the ocular damage. Okay, switching topics a little bit, I talked about upstream regulators, which are simple, let me just back up, simple networks like this, where there's one upstream regulator directly interacting with data set targets. But it's possible you can create higher level predictions based on, let me go through these again, a master regulator, or sort of a super upstream regulator, might work by activating or inhibiting other regulators, which in turn work directly to talk to the data set. So this is the idea of this causal network analysis, and it can find completely novel, potentially, master regulators, because there's not necessarily any literature that directly says, you know, this regulator increases the expression of that gene. But there's literature that says this master regulator may increase the activity of this transcription factor or this other protein, which in turn activates the expression of this gene. So that now you have a new hypothesis about a master regulator that controls a number of intermediate regulators. Another aspect of this tool that we have is you can say, well, I'm interested in master regulators that have a certain property. They connect to a disease or function or molecule of interest. I want to see which ones are like uber regulators that drive potentially directly or through even an intermediate drive some outcome. So that's where you can add a specific function. Like you could say, only show me master regulators that have a connection to encephalitis or to something else. So that's how this tool is set up, and the results, again, look like upstream regulator analysis where you get regulators and you get et cetera, but you can also filter for molecule type. And one of the things you can do is filter for compounds as master regulators. They don't have to be genes. So this could be this master regulator can be a compound that affects, inhibits some molecule or activates a molecule. So filtering for compounds as master regulators, there's a number of them, and this is again the z-score. So these are, and there's a negative z-score. So what these are compounds predicted to be inhibited, and the way we interpret that is that essentially, since the compounds aren't in the system, it's prediction that the compound, the expression pattern you're seeing in the data set is sort of the opposite of what you'd expect if that compound were added. So it's almost as if it's saying if you add this compound, you could reverse the effect of whatever system you're looking at. So this is one of the example one of the compounds that came out. This is xanthohumol. It's the master regulator. It talks to these data set these intermediate regulators. The data set molecules aren't even shown here because there could be many of them, hundreds. And these are, and then again the scoring. So we added the encephalitis as a output. Like how do you find a connection between the compound or any master regulator and the endpoint? And in this case, working through TLR4 or TNF, inhibiting those two, you could increase encephalitis. So it could be that this compound, by the absence of this compound, increases encephalitis. I'll explain that in a second. But there's a paper that talks about the neuroprotective effects of this compound in ischemic stroke. So it's kind of like maybe there's some known neurological value, therapeutic value for this compound in kind of damage in the brain, which is interesting. And so, but anyway, one thing you could do is say, well, okay, let's simulate what happens if we added xanthohumol. So you can, in the IPA, you can turn it around and say, I'm going to add or make this red, meaning add this compound, and now you can see how it would theoretically inhibit encephalitis, or either help stop it from happening or cure it. So anyway, this is the kind of thing you can do with this tool to find interesting possible therapeutic compounds. Okay. And here's another case. So the neuroprotection I thought was kind of interesting. So we put neuroprotection in as a endpoint as well, and you could find another compound that has, well, sort of a direct effect on encephalitis and a secondary connection, or slightly more distant connection to neuroprotection, and they're the opposite effect. So in other words, not having this compound decreases neuroprotection, not having this compound increases encephalitis. So again, theoretically, this compound added to the system would improve both, make it better in both cases. And this compound itself has known antiviral activity. So it could be interesting to look at. Okay, so another power of IPA is being able to compare data sets, or compare, and it's not just to compare data. Lots of tools can compare data sets. Excel can compare a data set, but the point is to take it up a level to the biology. How to compare the biology of two data sets. So this is another study. I mentioned earlier, it's a microarray analysis of these human RPE cells. The retinal pigment epithelium is in the eye, and this is using human cells. So they infected those cells just like same MOI of 1 in 24 hours. So they're very similar conditions. They're very similar conditions to the other study, but it's not RNA-seq, it's microarray, and it's different cell type. So if you look just straight at the data set genes that are significant, and we filtered to give them similar amounts of genes from the two studies. So in the case of the RNA-seq, I mentioned it was a fold change, absolute fold change of greater than 2, and an FDR less than 0.05, and that's around a little less than 1,000 genes. With the microarray, the pigment epithelial cells, the fold change absolute was greater than 1.3, and a p-value less than 0.05. So it gives a similar number of genes, and only about 250 are the same genes, and the rest are different. And you can see how if you just plot them against each other at a fold change level, there's not that great a correlation. There's a few that are very highly expressed in the two. I think this axis is the RNA-seq. They go up higher, 500 fold change. And actually, I cut off this scale. It actually goes higher. And then these are lower in microarray space. But anyway, not super similar looking here. But if you look at the biology, you see some amazing similarities. So this is looking at the causal networks comparing their causal networks. And so again, the orange, this is something you can do in IPA, which is do a comparison. You can have up to 20 comparisons, and you can actually do trending and all kinds of things, looking for, or clustering, looking for interesting patterns. But this is just 2, so there's not as much clustering that you'd want to do trending, obviously. But here's that xanthohumol and prediction. This is the macrophage, like I just talked about. Here's the causal network. And if you look at the same, if you just click on this square and to see what the same one came back for the microarray, it's almost identical. So it's nice to see that kind of confirmation in a totally different data set that this compound looks interesting. But it's not always going to be this case that they're the same genes. That's what the power of taking it to a biological level as opposed to a gene-by-gene comparison. So, for example, if you look at the functional predictions for the two studies, they look very similar. In fact, inhibition of virus, that function, they look really similar. But are they really driven by the same genes? Or does this have the same biology going on using slightly different gene sets? So this is what that shows. So this is the, anyway, this is the inhibition of virus. This title's wrong. It was supposed to be the inhibition of virus. And what you see is the red ones are the RPE ones. And these are were only changed in the RPE. They weren't changed in the macrophages. These were changed by both. And switching it slightly, then this is the ones for the macrophages. So you see they have a common set of genes, but they also use some different ones. So you capture more biology by looking at this higher level in IPA. And again, the yellow meaning there's multiple isoforms. And in fact, if you double click on any of these with multiple isoforms, like this yellow, there's a molecule called Z3-HAV1. So double click on that. It brings up a little summary of that molecule. And this is new, just in the last release, that shows the isoform level, a little preview or thumbnail of the isoform level data. So what this means is there are 4 isoforms known in Ensemble for this gene, 2 of which are actually changed in expression in the data set. One of them we choose as the fold change to represent the whole gene, and that's what's shown by this little check mark. But you can then simply click on this little section and go to a more detailed view of, okay, what's going on at the isoform level. So this is that part of the gene view that comes up, which before we did RefSeq support, now we also have Ensemble support. And you can see over here on the right, it shows the actual expression values for the isoforms that are in the data set. So it's in scientific notation, this 33 fold change for this one, this short form, which is kind of interesting. It's missing this piece. And if you look, that's actually a known, it has a name. That isoform has a name called ZAPS. And it's a potent stimulator of signaling mediated by RIG1 helicase for antiviral response. So that's kind of interesting. So this one's clearly, they're both upregulated. The two forms are both upregulated, but this is upregulated quite a bit more, 33 versus 7. And if you go back and say, okay, RIG1, so you see how the RIG1 pathway had come up before. And in fact, if you go back and look at the causal network analysis, you also see that the ZC3-HAV1 is itself a causal master regulator, and partly by working through RIG1, which is also called DDX58, to drive expression change in the data set. So this is clearly an important antiviral molecule that comes up. And it has a, has isoform level information that's interesting. Okay, so I am going to stop and just summarize by saying that I hope you found that IPA is useful to study host response to infectious disease. It allows us, to explore the viral disease mechanisms and even potentially find therapeutics, look at pathways, upstream regulators. They can be targets. They can be therapeutics themselves. You can really understand at a molecular level hypotheses around host genes and how they respond to the viral infection. In fact, there are 500 papers more that use IPA for infectious disease. And we're having a promotion, so you ring in the new year by getting an advanced analytics license, and you get one free, or you can get a 25% discount. And advanced analytics, what that is if you don't already have it on your IPA license, it allows you to do the causal network analysis, which I talked at length about. It also gives you Bioprofiler, which is I didn't show, but it's a tool to look at detailed relationships between genes and compounds and diseases and functions in a really detailed way, which genes are causal for a disease, things like that. You can, and you also allows you to do relationship exports. You can export relationships from networks. So anyway, that's available if you buy it by March 31st this year. Contact your account manager or call customer support, and you can mention this promotion and get that discount or the free one with the single license. And at that point, I'll stop here. Take any questions, and again, I also have Jean-Noel and Sean on the phone for other questions.
Thanks, Stuart. So those of you who have any questions, please use your question box and enter them in. It can be for Stuart, Jean-Noel, or Sean on the line. I think we are good. So thank you so much, Stuart, for your time, and oh, hold on. Oh, I just didn't refresh. Says, I wonder, the RNA-seq data shown in this nice presentation mostly relates to host responses. Does IPA also have a good coverage of bacterial information?
No, good question. IPA is oriented around human, mouse, and rat literature. So, there are findings that relate to how a bacteria affects human, mouse, or rat, but there's not specific, if there's information that's just about a bacteria, it wouldn't end up in our knowledge base.
Okay, next question. In IPA, can I cancel the yellow circle, parens isoform?
Can you hide them or get rid of them?
Mm-hmm.
Yeah. So actually, I can show you that. Oops. Can you see my screen, including IPA?
Yes.
Michelle? Okay. Okay. Where do I want to do that? You would go to the let's just open a pathway just to show you. Let's open our little friend, interferon signaling. So what it is, it's an overlay. It's considered an overlay. So what you do is you go to the overlay menu, and then you go to the analyses, data sets, and lists menu, because this is overlaying the data set information. So that display isoform information is the checkbox you want to check on or off. The other thing you can do is, if you don't like the pink, there's also a pink highlight because it's sort of emphasizing which molecules came out of your data set. You can also reset that highlight as well, so you get rid of those pink or fuchsia, I don't know what the color is, circles around the genes. You can then do that. So this is like the simplest, cleanest look of it, where it's just the expression changes without any other information.
Thanks, Stuart. Next question is, can we do metabolic analysis using IPA?
Indeed, you can. You can upload a chemical list, an endogenous metabolite chemical list. An IPA should recognize those identifiers, and you have to, or you can use the name of them. But anyway, upload a list of chemicals, and then it'll do an analysis of those, try to find out which proteins they interact with, which metabolic pathways they fall into, and things like that. So there's an actual. After you upload that data set, it's called the metabolomics analysis. So absolutely. You can also mix together compounds and genes. If you happen to measure gene expression on the same tissue, on a tissue, and then you happen to have the chemical analysis of that same tissue, you can upload them together in one data set. So.
Thanks. Okay, next question is, can you export networks to other programs like Cytoscape so you can more easily play around with the layout?
Yeah, so again, as I said at the end, if you have an advanced analytics license, and there is that promotion running right now, yes, you can do that. You go to export. Once you have this license, what I'm about to show you won't work unless you have upgraded your IPA license to have advanced analytics, but then you get all relationships, and then you just say okay, and it'll export a pairwise relationships, A binds B, that kind of pairwise, or triples, as you sometimes might call them. Export them into an Excel file or text file, and then you can upload that directly into Cytoscape. It's very easy to do.
Okay, next question is, in IPA, what are the white circled regular genes?
White circled regular genes. I guess maybe you mean like these genes that aren't colored. I'm not sure. I'll answer it that way, and then if you need more if you want to clarify, that's fine. So these are just genes that are in the pathway that aren't in your data set, or that didn't pass your analysis. That aren't in your data set. So SOX1 wasn't in this data set. That's why it's white. Gray ones are ones that are in the data set but didn't pass your analysis cutoffs. So like I said, we filtered this to fold change just greater than 1. So this has a fold change greater than 1, but it didn't meet either the p-value cutoff of 0.05, FDR of 0.05, or it didn't meet the cutoff of fold change 2.
And that covered the next one, which is what was the gray. Next question, if I'm analyzing a rat microarray data, should I expand my analysis to include human and mouse knowledge base?
Yeah, that's a great question. As I said, we sort of integrate the 3 species together by default. And I prefer to run everything at first with all species, because what's found in a mouse is probably relevant to what's found in a rat, and what's found in a human is often relevant to what's found in a rat. So you start out with all 3 species by default, just run an analysis, or do whatever you want to do with all 3 species. And then if you want to make sure it only occurs in rat, then then strip it back down to just showing rat, because you can filter just to rat. You know, when you set up an analysis or whatever you do in IPA, you can always filter down just to rat. But again, starting with everything, get the full picture. Maybe you get an idea that you wouldn't have gotten if you just look at rat alone. So.
Thank you. Next question: what underlying algorithm are you using for the isoform modeling?
I don't quite understand that, but I mean, in this example, we use the ensemble gene model, but we have, IPA also understands the RefSeq gene model. You can switch it back and forth. So let me show you an example of that. Like if I open stat2, it's going to show you this isoform section. This one has a lot of isoforms, and some of them are expressed. They're ordered from largest to smallest, not that you can really tell in this view, but it's just so you know how it's ordered. And you can get a little hover as to what they are. But again, if you click through to the gene view, it shows the results in ensemble space by default, because my data set is in an ensemble space. And so it shows, you know, the different ensemble-based splice information, and indeed it also tells you what the support level is from ensemble. So if you know ensemble, it tells you it's, you know, biotype is protein coding. It's a TSL1, meaning it's really strongly supported by, you know, expression data or genomics data. You can see some of them have less support. We don't editorialize. We're taking in what ensemble has, and then we just tell you what support level it is. And the workbench, you know, we use the CLC workbench in this case. It used the same model, so it maps the fold changes to the same genes or transcripts. I mean, again, though, you can switch it. So if you think I really care about RefSeq, this is the RefSeq version, and this doesn't. RefSeq in our case doesn't include XM or predicted transcripts. So it's usually much fewer in RefSeq space than ensemble. I hope that answered the question.
I'm sure it can shoot over another one if it didn't. Next question is, can you show a way in which I can upload a list of genes of interest in the search box?
Oh, sure. You don't upload it per se, but you can copy and paste. And I don't want to try to do that right now, but if I had gone to Excel or someplace else where I had a list of genes, you just paste them in as a comma delimited. So, you know, and I think if you paste just like a list that's return delimited, I think it'll just automatically comma delimit. I'm not sure. But anyway, you just paste it in here. Imagine I pasted that, and then you just search, and it, you know, it just does a OR search for all of those.
Thank you. In the simulation method, can you add or inhibit multiple genes and see their network effects at once?
Sorry, I spaced out for a second. Can you read that? Say that one more time?
In the simulation method, can you add or inhibit multiple genes to see their network effects at once, all at once?
Absolutely, yeah. So let me see if I can show you that really quick. Yeah, so let me see. Basically, it's in the overlay tool. It's called the Molecular Activity Predictor. You can clear it, you know, clear off everything that's there, and then you can just go ahead and use this paint bucket, which is sort of a thing like, so it says, you know, the red ones meaning it's upregulated or activated, and you can click multiple, you know, you can click multiple guys. And then, oh, sorry, I didn't turn on the prediction yet, but you can hit start prediction, and it'll go ahead and use all the molecules you set. And you can set them in different states, too. Like, what if this molecule were green, inhibited, you know? So anyway, you can, yeah, absolutely.
Great. And last question, unless any others pop in. In this seminar, you compared RNA-seq with microarray data, both at the RNA level. Can we compare, for example, RNA data with proteomics data, or even one level higher, compare RNA data with phenotypic data, for example?
Yeah. The phenotypic data, I can't quite think of how you would do that, but definitely the RNA or protein, you can have a protein data set. Let's say it's fold changes of protein expression. Absolutely works just fine. And you can create an analysis in IPA with that level of data, and then you can just do a comparison like I showed. Yeah, absolutely, that works. Phenotypic data is a little different because you can't exactly import phenotype data like blood pressure 140 or something. There's no way to import that exactly in IPA like that. You could always search for those kinds of things and then see what their effect is. Simulate effects with genes and things like that, but you can't upload a data set of like clinical data or something like that.
And then we got another question in. Is there a way to display a key for the degree of coloration in data set molecules?
Is there a key? No, that's something that we've been asked to do, and we will at some point. But it's at this, we don't have a key there's a key for the colors, red versus green. So and you can change that. So if you go to preferences, you can, this is what these mean. But there's not a scale that says, bright red is 12 fold up and dark green is -4. We don't have a scale like that in a given analysis like this.
Thanks, Stuart. And I apologize to one of the attendees. I accidentally deleted your question. So if you just recently submitted a question and we haven't answered it, I apologize. If you would resubmit, we can answer it. Otherwise, we'll be closing. So I just want to give a couple seconds because I hit the wrong button and there was one more question. So if your question hasn't been answered, aha, here we go. Can you insert data from literature to get a comprehensive view of all the knowledge of a field and compare your results to it?
That's a good one. So we have an additional feature called My Findings, which exactly enables that. So essentially, either on the fly, or and what I mean by that is like right now, let's say I decided that I wanted to add something to this pathway. You can add a particular molecule and a finding that's only yours, that only you know about. You can add it to a pathway or to add, and it goes into your knowledge base for your license. So if you're at a particular company or something like that, it's only your people on your license, your company can see it. You can upload it, you can do it one by one like that, or there's a batch tool where you can create a sort of simplified format in a spreadsheet of findings that you've gotten from the literature and upload them all at once, thousands of them, and then use them in your analysis, use them, and everyone else gets to see them, and you can organize them into projects. So let's say you have the cancer findings project and you have the asthma finding project. You can put those into separate projects and use them just like you would any other source of findings in IPA, and it's only for your company. So if you're interested in that, contact your account rep for that, or account manager for that.
Okay, and we've got a couple more sneaking in here. We have a little bit of time left. There's a cited article from the Journal of Investigative Dermatology. Can I see this? Oh, slide for the article.
Can you see the what for the article?
The slide for the article from the presentation. But I'll be sending out this video recording in a few days so everybody can see it again on demand. And then if I want to compare two data sets, is it necessary to set filter sets so that I can have similar number of genes to analyze in both data sets?
No, in fact, that kind of came out. I didn't purposely try to make them the same. Anyway, that's what happened. But no, you don't have to. In fact, I usually would set, especially if they're done in a similar, if you have sort of a similar set of experiments, I would almost try to pick one set of cutoffs that you use globally for all of those. So in other words, let's say it's 1.5-fold change and p-value 0.05. I would use that same setting across everything and then see how it comes out. Some may only have 200 genes that pass that cutoff, and some might have 2,000 that might pass that cutoff. But I wouldn't necessarily try to make a match. It kind of happened that way in this case, but I didn't really try to do that.
All right, well, that takes us to the top of the hour almost. Stuart, thank you very much, and Jean-Noel and Sean for joining us, and also to all of you who attended today. This will be recorded or has been recorded, and I will be sending it out in a few days so that you can watch it again on demand at your leisure. Thanks again, everyone.
Thanks, Michelle. Thanks, everyone.